Membranes are composed of largely of lipids and proteins. This is basically the model of a membrane that is largely accepted as the most accurate model is referred to as the fluid mosaic model, which contains predominantly a phospholipid bilayer, which also contains some cholesterol, and then proteins that are inherently part of that bilayer. Generally, these proteins come in a different variety. If we look at a couple of these examples, this right here would be an example of a transmembrane protein because it goes all the way across the membrane. Whereas this particular protein over here is only associated with one side of the membrane. This would actually be the outside portion of the membrane. The little blue things attached there are carbohydrates, and carbohydrates are always found on the outsides of membranes. Basically, the proteins that are actually embedded within the membrane can actually be a very large part, sometimes as much as 50% of the weight of a membrane, depending upon the particular cell. And a lot of time those there are very specific functions that are played by each of those proteins. For example, if a protein like the one that I've highlighted here has, you'll notice that there are several different alpha helical domains in this protein. More than likely, there's a channel that is present in the interior that allows things to pass through that interior channel. So this would probably be some sort of transport protein that's meant to get things from one side of the membrane to the other. We also have proteins that serve functions for cell signaling, allowing signals to cross a membrane, as well as allowing things to attach to the membrane. So if we look at the composition of the phospholipids, basically we saw earlier how phospholipids have a phosphate head group and a nonpolar region that's composed of fatty acid tails. In general, we can look at the importance of both unsaturated and saturated fatty acids within a membrane because the saturated fatty acids tend to tend to decrease the membrane fluidity, whereas unsaturated fatty acids would increase it. Just like a saturated fat and a triacylglycerol basically makes for a very solid something like butter or lard, whereas something with a lot of unsaturated fatty acids would tend to increase the fluidity, make an oil essentially out of a lipid. Cholesterol plays a very important role as well in that if we look at the lower figure here, here's a molecule of cholesterol that has inserted into the membrane. Notice that the hydroxyl group is associated with the phosphate portion. The rest of it inserts itself into the membrane. Generally, cholesterol can increase the rigidity of a membrane, but there are certain aspects to cholesterol that can actually not increase the rigidity as much as a saturated fat would. And largely in animal membranes, simply by controlling the amount of cholesterol that's present in the membrane, we can very often control how rigid a membrane happens to be. Plants, on the other hand, tend to use more unsaturated, putting more unsaturated fatty acids into a membrane to increase the fluidity of a membrane. Membranes are selectively permeable, meaning that only certain things can cross a membrane. Other things that cross a membrane need to have some sort of transport protein. So, for example, ions, which have a positive or negative charge associated with them, do not cross membranes very well at all because the hydrophobic interior of the membrane is very resistant to movement of charged molecules across that membrane. They would instead need to be transported by some sort of carrier protein or some sort of channel in the membrane. Molecules like carbohydrates are very hydrophilic in nature, and thus don't move very effectively through the lipid portion of the membrane. So generally we need some sort of transport protein for those as well. Nonpolar substances, on the other hand, tend to diffuse very easily through a membrane. And this can include things like gases. Oxygen and carbon dioxide, for example, move very effectively through membranes, as well as various molecules that happen to be lipid soluble. So a lot of our drugs that are used generally tend to be rather hydrophobic molecules, so they can very easily cross a membrane in order to allow them to get inside a cell without requiring some sort of carrier protein. Now, the phospholipid present in a membrane gives certain aspects to a membrane. The first of these being the self-healing capacity, because if you break open a membrane, you expose the hydrocarbon tails of the phospholipids to an aqueous environment. And as a result, this is, we've talked about the hydrophobic effect with regard to protein structure. Same thing happens with phospholipids. Hydrophobic effect is very important in making sure that the phospholipid tails stay in the interior of the membrane. So when a membrane is broken and the tails of the hydrocarbon tails are exposed to the aqueous environment, they naturally want to go back into the typical membrane form to prevent that interaction from occurring. Phospholipid membranes tend to be asymmetric. Generally, if you look at the exterior portion of a membrane, it's going to look much different in terms of composition from the interior, and we can actually tell something about membranes by the actual compositions. In addition, we can very often tell when a cell is under stress by noticing certain phospholipids that might be present on one leaflet of the membrane when they're not actually supposed to be there. Membrane proteins, as I said, can sometimes be as much as 50% of the weight of a protein, and they come in a couple of different varieties. There are integral membrane proteins and peripheral membrane proteins, and basically the peripheral membrane proteins are kind of the easiest. Basically, they simply associate with the phospholipid heads, largely through things like electrostatic interactions, possibly some hydrogen bonding. Basically, peripheral membrane proteins can be very easily removed from a membrane, whereas the integral membrane proteins are relatively tightly bound to a membrane. They are either partially or fully embedded within the lipid membrane. The protein label number 2 here would be fully embedded, and actually it would move through the membrane by basically moving through the lipid tails. This protein here obviously represents an entire transmembrane protein, since it goes all the way across the membrane from one side to another, and then this one is only associated with one side. Now, in terms of the membrane or the makeup of these proteins, the protein labeled number 2 here would have a lot of hydrophobic amino acids on the exterior portion of the protein, basically to allow it to interact with the hydrophobic regions of the phospholipids. The interior of the protein would probably have a lot of hydrogen bonding and electrostatic interactions occurring, such that because the hydrophobic forces would not really be a driving factor for the proper folding of such a protein. So basically kind of the inverse of a typical globular protein. Looking at proteins 1 and 4 here, there would probably be a lot of hydrophobic amino acids that would be on the portions associated with the membrane, whereas these regions of the protein would have a lot of hydrophilic amino acids for association with the aqueous surroundings of the membrane. There are a couple other ways to anchor proteins add a membrane. One example is by attaching a fatty acid. So here we have a fatty protein where the N-terminus reacts with a fatty acid basically to form an amide bond, and the fatty acid is actually then stuck into the membrane, and this kind of sequesters the protein at the membrane. Additionally, we can add isoprenoid type groups. What we see here is the example of farnesylation, adding a farnesyl group to a protein. In this case, this is added to a cysteine residue, and what we see there is the formation of what's called a thioether. It's essentially an ether that instead of having oxygen in it, has a sulfur atom in it. And once again, the farnesyl group would then just insert itself into the membrane, and then it would pretty much the molecule would be stuck there. Lipid anchors on membrane-bound or membrane-associated proteins actually make these proteins relatively difficult to remove because of the hydrophobic interactions that take place between the lipid anchor and the particular protein in question.